Hey, this is Colin McNamara from Austin LangChain. I want to introduce Venika Kakarla's presentation she gave in our March meeting, where she basically found a way to detect the cancer that killed her grandfather in really interesting ways using AI and ML. So without further ado, Venika's presentation.
Hello, good morning. My name is Venika Kakarla, and I'm a sophomore at Round Rock High School. I've had a great interest in AI since I was about 14, and most importantly, in the intersections of AI with molecular biology and healthcare. I decided to do my project on AI usage in cancer research, which is a vast field that is still unexplored. And by utilizing the power of these advanced systems to detect similarities unseen to the human eye, I believe we can discover new patterns and similarities in cancer to help researchers and doctors understand these different and complex cancers even more. So most importantly, what I'm focusing on is a cancer called hepatocellular carcinoma, or HCC for short. And this is a liver cancer, a fatal liver cancer that has many different factors contributing to its fast cancer growth. Furthermore, it's a very complex cancer, which makes it so that it is greatly differs from patient to patient. This is quite differently caused by a viral infection, which is not as common in cancers. However, this viral infection can cause mutations in DNA sequences in the TP53 gene, which therefore causes mutations in a protein called p53, and this is vital for tumor suppression in tumor growth and in hepatocellular carcinoma. Now, why is p53 important in this case? So this protein helps regulate the cell cycle, which controls how fast tumors can grow during this cancer, and it pretty much determines how much time a person can live for, or what's the rate at which health is going to deteriorate. So mutations in this protein, such as even like single amino acid changes in this protein, can affect the way it binds to DNA and how it can control the growth of tumors. So the problem here is that now that we know the biology, let's discuss the problem at hand affecting millions around the world. So this is the third leading cause of cancer-related deaths worldwide, and the varying nature of this cancer makes it very difficult for researchers and doctors alike to diagnose or record the growth of. However, the small protein p53 that I was talking about, more specifically its DNA binding region, which includes its main residues, provide critical insight into tackling the unpredictable nature of hepatocellular carcinoma. Approximately 50% of patients worldwide with HCC have mutations in p53, and this protein is vital to controlling the growth of cancer cells. But without its function, the mutated cancer cells will be able to replicate without bounds. And with the addition of a single amino acid change in the DNA binding domain, the stability and binding affinity of this protein to promoter sequences of DNA that facilitate tumor growth could be altered, leading to more aggressive cancer proliferation. To counter this vital problem present in HCC, I conducted a comprehensive study on p53 point mutations and developed a novel machine learning model to understand the mutations' impact on stability and DNA binding affinity to properly identify and understand patterns in this cancer. Of course, there are many current solutions that p53 stability and DNA binding affinity. However, a lot of these solutions are either costly or tedious and usually done in labs, which not everyone may have access to. And most importantly, ATC does vary very much between patients, as it is determined by many different factors, including the different mutations that can occur in p53. So the current problem arises with costly and accessible simulations taking hours of time to predict the stability and DNA binding affinity of one mutant protein. For example, labs can cost upwards of $10,000 just to predict the thermostability and binding affinity for just a few of these proteins. And this kind of money is not available to all researchers around the world, and it limits the advances of this research. Although scientists have created machine learning models as well to efficiently predict stability, these models are trained on many different proteins with greatly different structures and may not guarantee p53 specific accuracy. Furthermore, almost no machine learning models can or have attempted to predict p53 DNA binding affinity, which is a serious problem, as this is what pretty much determines how fast the cancer can spread. And this inefficient analysis of the p53 reliant HCC contributes to slow diagnosis time and an incomplete understanding between patients. In order to combat the inefficient analysis, understanding the prominent patterns in the protein structure would prove vital to proper understanding of its functional capabilities as well. So creating a way to predict the thermostability and DNA binding affinity, utilizing specificity, efficiency, and a novel prediction method is vital to trying to figure out how this cancer spreads in humans from patient to patient. And this is why I created a machine learning model that can analyze mutant p53 stabilities and binding affinities with DNA, utilizing patient-specific amino acid sequences. This combines specificity to p53's structural and functional aspect prediction, efficiency, and a novel prediction method to predict changes in binding affinity, specifically from wild type to non-mutated or to mutated p53. So for my initial analysis, I took about 1,400 cases of mutated p53 protein from the COSMIC database, which is a publicly available database, with each sample showing the point mutation in the amino acid sequence. I then pre-processed it to only include significant point mutations without repetitions, and that came out to be 249 cases. And utilizing Python scripting, I then applied the mutation to the wild type p53 sequence, and then the DNA binding domain was extracted and stored so that it could be processed through AlphaFold, which is a protein structure prediction software. And those protein structures were saved so that I could analyze them and use them to create a machine learning model. Furthermore, I also utilized scripting to create adjacency matrices, and those are just Excel files which show the interactions between residues of a p53. And another Excel file I used to show the interactions between the p53 and the DNA. So these are just some of the examples of the data files that I use. So the COSMIC database sample in Excel, it shows all of the different cases. So it shows like where they found a tumor in this person and what mutations were present in that p53 gene in this person. And those were 1,400 samples. This is just a sample of that. And the mutant adjacency matrix file, I made 249 of those mutant adjacency matrix files for each of the different mutant proteins that I had. And those just shows the distances between each of the residues. And I also had a file that shows each of the mutants' structural features, such as hydrogen bonds or salt bridges, and it records whether the mutation was in which region of the protein. And this included about 249 rows. And the DNA adjacency matrix, this is just a sample of it. And this shows the interactions between the p53 residues and the DNA. And the ones represent interacting residues, though that means just like it forms bonds between the two structures, and the zeros represent non-interacting residues. So this was my machine learning model framework, and I utilized 3 different algorithms to create this. So I first utilized a deep neural network, and the data that was processed, it was first inputted into the deep neural network, and this had many layers to output whether the p53 DNA binding domain was stable or not. And I also utilized an XGBoost model for regression to predict the numerical stability value of the p53 DNA binding domain. And finally, those 2 inputs were also put into a graph conventional model, And this was able to see a graph-based input of the protein structure, which is more specific to what P53 looks like. So it was able to analyze this in more depth than the previous two models and was therefore able to output the interaction probability of a mutant P53 DNA binding domain CDK1A sequence, and that's just a type of gene that I utilized as a percentage of the wild type, and wild type just means like non-mutated P53 interaction. And more specifically, to go over the different algorithms that I use. So the deep neural network for the input layer, each neuron in the input layer represents a feature from the data set, such as hydrogen bond count, solvent accessible surface area, and other encoded values. Each feature is then connected to all neurons in the next layer, where this raw data is passed to the first hidden layer. And then the feed forward function is utilized in each layer to apply a transformation and produce an output that will be passed forward. The activation function, which is just like a nonlinearization function, analyzes whether the mutation buries or exposes a residue with basic feature interaction analysis. And then the second hidden layer allows for more complex interactions than the first, and it analyzes different regions of the protein and mutation occurrences in such domains with weighted neurons. And finally, the dropout layer eliminates neurons that, which is crucial to prevent overfitting, and instead supports like generalization of the data. Finally, the output layer gives a probability utilizing the sigmoid function in binary cross entropy loss, so that it can predict whether the mutation was stabilizing or destabilizing. And this output is then passed to the XGBoost model for regression. And it utilizes decision trees that learn patterns and improve over time to make proper predictions. A loss function to predict the mean squared error is included to compute the results along with the gradient boosting equation for the trees to be properly optimized. In order to predict numerical thermostability, first tree is a high-level split from the input. So it utilizes the input data from the DNN, and that's the first classification. The second utilizes solvent accessible surface area, which is a feature that is vital in understanding the thermostability changes. And then the third analyzes hydrogen bonds, determines stability values based off of that. And the fourth and ones after that utilize Saltbridge's mutation position and residue type to refine like stability predictions, properly output stability values. And this input is then fed to the graph conventional model to predict DNA binding affinity. So this is more of the novel part of my project, as this is able to predict DNA binding affinity, which has not been done before and is quite complex. And after receiving the numerical thermal stability value from the XGBoost, the GCN will convert the adjacency matrices into graph-based inputs that show the features of the edges and nodes. And edges are just the interactions between these different residues of the protein, and nodes just show the different structural features, such as the accessible solvent area. And the local feature learning or convolutional layer uses ReLU to show nonlinearity, and it emphasizes the individual residue interactions with the surrounding residues. The second layer also uses the ReLU function to expand the residues surrounding to non-direct neighbors while encoding the effects created by the mutation. And the pooling layer puts together the wild type complex and the mutation that it is analyzing to finally output the interaction probability that this mutant P53 would bind to that DNA sequence using the wild type complex as a base. And from my results and analysis, I found that after running many tests, I found from the two best tests of each, the DNN and the XGBoost, about a 96% accuracy on 50 epochs. And that's just how many times it runs through the training. And on the XGBoost, I found a 99% accuracy, as it was able to predict the thermostability values quite well. And furthermore, for my results and analysis, these two graphs on the left, they just show the validation results from the two different models. So the regression one is the XGBoost one, and the DNN just shows the training and validation loss for the 50 epochs. And then in the middle, I have the mutation interaction probability. So this was what was outputted for those 249 mutations from the GCN. And this just shows all of the different probabilities that the mutant P53 would interact with the DNA. And as you can see, most of these are very low in the 20s or 30s range. And this means that the mutation probably greatly affects the structure and function of the protein. And this was able to predict it for all 249 of those mutants. And this was from an initial analysis result, the frequency of point mutations. That was from the initial analysis of the database, and that just showed a certain mutant appearing most often, which I will go over. And this shows quite a negative correlation, just a little bit of a negative correlation between binding probability and thermostability, as the model did use that as an input to predict the binding probability. So what I could conclude from this was that this is the first P53 specific machine learning model that is able to predict stability and binding affinity of a single amino acid change in the P53 DNA binding with domain in just a few minutes. Usually the current solutions that are used today, if I were to utilize a computational model or like molecular dynamics simulations, it would take about 2,400 hours, 2,500 hours to predict all of these values. However, this model was able to predict those 249 mutant values in about 3 minutes. So this is quite an improvement. And utilizing the initial analysis data, I also found that more on the scientific side of things, that an R249S mutation, which is an arginine to a serine, switched that amino acid and decrease the stability, therefore decreasing the binding interaction probability. And I found statistical majority of P53 related point mutations affect the DNA interaction. This makes them have a lower interaction with the DNA and more fast cancer progression. So this shows that even like a small mutation in this protein can lead to increased proliferation and worsen cancer progression. And furthermore, this tool can save researchers a lot of time and is novel to understanding this protein. However, it can also be used in more than just hepatocellular carcinoma. It can be used for many different cancers, as this protein may be mutated in a lot of different cancers, and it is a main protein in many different types of cancers, and it controls the cell cycle, which is a main part of all different types of cancer. So it has a wide variety of uses. And for my future work, I am planning to gather more data about this, maybe in a lab setting, and gather data on the DNA binding interactions in a lab setting so that I can get more accurate results and then verify it as well with the GCN. And then I will consult with professionals, which is what I'm doing right now, and so that I can understand more about the machine learning part of this. And then I will create a web-based interface that allows researchers to utilize this tool so that they can understand more about the interactions of P53. And then finally, I hope to upscale this model so that it can be used by researchers worldwide, and then they will be able to learn more about this cancer and many other cancers. I would like to thank Mr. Brian Port and Mr. Colin McNamara for giving me this opportunity to present, and thank you.
See if I can get this microphone working. Thank you, Venika, for your presentation. So a couple of questions, and Brandon here, if you want to have any questions from the audience, but I'll leave one off. First off, wow, this is really, really great stuff. It actually reminds me of friends, machine learning research in Silicon Valley right now. And, he's sold his 5th company. Oh, and you would be impressed by what you're doing here. So what was your inspiration for applying machine learning to the specific field?
After I realized how machine learning and AI can help us understand more about different cancers and help researchers mostly understand more about like the patterns in this cancer and how specific elements of these different cancers can affect the growth or tumor growth. I decided to try and apply all of these factors into like a machine learning or AI model so that it can accurately predict patterns that maybe we were not able to see or different researchers were not able to see, so that we can understand more about this. And my inspiration behind this specific topic was a family member of mine actually had this cancer and passed away from it. So I wanted to learn more about it and try to create an impact in this field so that I can change the lives of many in the future, hopefully, so that they don't meet same fates.
Very inspirational. How many people around the world about stomach cancer?
This cancer, hepatocellular carcinoma, is actually widespread. It is actually the third leading cause of cancer deaths worldwide, and it's very fatal, and it's growing even more because it's usually caused like by bad lifestyles or unhealthy, like unsustainable like environments. And yeah, many people are affected worldwide. It's the third leading cause of cancer deaths. I don't know the exact number, but I know it's more prominent in rural countries or in third world countries, and but it is still prominent in all different types of countries.
So with the reduction in machine hours necessary to detect this type of cancer, like how much money does it, or how many US dollars does it take to run a model like this and detect it?
So actually, I created this model on Google Colab, and I started about yeah, it's still like a prototype model. It's not like to be used in like a professional setting, but it is able to predict the binding affinities pretty well. And the GCN is actually really useful for analyzing all of the results and creating like a more graph-based input, which is what a protein would look like. And that really helped me to understand like more about this protein and allows the model itself to understand more about the protein. So yeah, I'm pretty sure this would help more people gain access to this kind of research and hopefully advance this cancer research in the future.
So there's a bunch of researchers, a bunch of scientists. Who uses Colab, Jupyter Notebooks? Like, yeah. Hey, so some of our first labs were hosted in Colab to run AI applications. Like, sweet, give us free compute for $10 compute. What questions we have from the audience? We have a microphone in the back. I think that's Constantine.
Okay, yeah, first of all, really interesting work. Second of all, I'm curious, why did you pick
So actually, I was planning on using a support vector machine at first, which is more widely used for thermostability predictions. However, I wanted to see how different parts of this can predict like different parts of thermostability. So I wanted to break it up into two different models, which is an XGBoost, which is like a regression type model, and the deep neural network. So this would be able to, so if like even it fails in predicting whether it's stabilizing or destabilizing, it would still be able to predict like a proper numerical stability value, or the two wouldn't interfere with each other too much. And so it reduces like the error over like a traditional support vector machine that's just like one model to predict both of these values. So I decided to use that architecture for the XGBoost and DNN. And for the graph conventional network, usually those kinds of models like graph-based models are more widely used in protein analysis using AI. And those like are able to show the computer or the model what the interaction between the different residues are. So that would be more useful for the algorithm itself to see the interactions. So, mhm.
Report verification, My first question is, how do you code it?
So I use Google Colab to code it. I use many different libraries like TensorFlow and Torch Geometric, which is used for the GCN. And I started learning AI and machine learning a while ago, so I know a bit about coding it. The only newish part to me was learning about the graph conventional network. And this was actually one of the first like big projects I did with AI. So I spent quite a while on this, about 6 months to learn all of this, to learn how to code this and all. And I watched a bunch of tutorials and learned about these models, how they can be utilized. And yeah, I just started working on it in Google Colab, ran into a lot of errors. I did use AI to help me understand my project, so understand like the science behind it and all. Whenever I ran into errors, like I wrote the code myself. However, I did use AI to understand like what went wrong or how I could fix these errors. But yeah.
What was the significance of that data you took?
Yeah, so hydrogen bonds, solvent accessible surface area, those were some of the features I had. And those are main features in proteins. So for example, like hydrogen, like how protein is, like if there are more hydrogen bonds, then it is probably meaning that like the protein gets denatured or destroyed at higher temperatures. So it's more likely to be able to function in the cell and control tumor growth and all. And yeah, most of those are more biology related. And yeah, I learned about those during like my biology classes and all. So I was able to utilize that knowledge and pick these different features that determine like the stability of a protein, how well it can function. And one of the central themes of biology is that structure determines function. So I was able to incorporate these different features that determine structure as well as function. So I was able to use that to be inputted into this model and properly predict the different like stability values and all of that.
Cool.
Yeah, both of those processes took quite a while. A lot of the machine learning part, it was just like trying to fix errors, trying to figure out what went wrong, how I could improve things. I ran like a lot of tests trying to change the hyperparameters and all. And yeah, about 2 or 3 months I spent trying to understand all of the different aspects of this, like about the proteins and how it affects tumor growth. And I read a lot of research papers on the AI part of it as well, of how they use different AI models to predict like the thermostability, and how I could use my own like novel idea to predict the DNA binding affinity of these different complexes with the different DNA sequences and the different mutant proteins. And yeah, that took quite a lot of time to figure out and put together into a comprehensive like report and model.
Yeah. So really cool. Congrats.
Thank you.
Any questions? Well, thank you very much for your presentation, the inspiration. You know, a lot of us deal in the middleware layer and experiment training models and stuff like that. So it's really great to hear you describe not only the story of the spark of your grandfather getting cancer and how that drove, you know, this very innovative, both in the research side, but I really think that something that I saw you kind of shrink a little bit on, which was the Google Colab. That is one of the most brilliant things out there. So you did this on almost zero resources. Like,
Yeah.
well, a lot of us, you know, if we're doing this work, might literally burn tokens on anything. You did amazing work with great results on zero resources. So terribly impressive. Thank you so much for sharing this with us today.
Thank you.
You're welcome.